Okay, so I'm gonna talk today about how you can use FCS to study the glucocorticoid receptor. I choose the glucocorticoid receptor because Diego talked a lot about this receptor and well, he simplified my life. So first of all, let me take some minutes to explain the idea behind the experiments I will show you and the idea behind most of the techniques you are studying or learning in this course. So the idea is the following. Let's imagine that you want to understand how a society is organized. So you can do two different kinds of experiments. The first one is just, for example, take this woman here. You can isolate her, purify her, put her in an artificial environment, for example, a test tube, and study what she does. The other possibility is observing how this woman behaves in her natural environment. For example, interacting with other women, interacting with other people, moving around. And from this kind of experiments, you can really learn a lot about how a society is organized. And I would say you can learn much more than in these simple experiments we used to do in the past. So the idea is observing cells, real cells, biomolecules moving around the cells, and learning about the cell's organization from this kind of experiments. So today, my woman will be the glucocorticoid receptor. Diego talked a lot about this transcription factor. And the idea is I will try to show you some of the experiments we did. I don't have the time to show you the whole story, so I will focus in a couple of experiments we did to answer these two questions. The first one is, can we observe the GR in action as the woman that moved in the movie before? Can we dissect its interaction with other biomolecules that are relevant to its function? And okay, I hope, Diego, you don't mind, but I took your drawing from your talk. So this is the wild type receptor, and in some of the experiments, I will show the comparison with the monomeric version of the receptor that, as you already know, it has 2-point mutation, and as a consequence, this receptor is not able to dimerize. Of course, we studied a lot of the mutations that Diego showed you before, but I will only focus on that just because this is a pedagogical talk. So how can we study the motion of the receptor or of any transcription factor in the cell? We are going to use fluorescence correlation spectroscopy. I think you learned a lot in the morning, but just a brief overview of the technique. Here you have, in this case, is a confocal microscope. You place on top of the stage a sample, let's say cells expressing a transcription factor fused to a fluorescent protein. And what you are going to do is focus the laser in one spot. We are going to do the most simple experiment of FCS, which is point FCS. So whenever you focus the laser in that point, in that small point, what you define is a small observation volume, a femtoliter side observation volume. So molecules are there in the sample. And what we are going to do is acquire the intensity as a function of time by using a frequency at time, if you want the sampling time, in the order of microseconds. So if we do that, what we are going to collect is the following. Every time a molecule goes into this observation volume, it will get excited, it will start fluorescing, fluoresce until it moves out. So if you register the intensity as a function of time with enough time resolution, what you are going to see is the fluctuations of the molecules moving in and out of the volume. So I believe that you already knew that, but doesn't matter. So this is a simple case. You have molecules moving in and out, you get the fluctuation, but let's imagine that now what you have in there are, for example, target sites. So molecules will come into the observation volume. They have a probability of attaching to this target site. So they are going to stand there for a while until they move out. So now you can see that this fluctuation will contain information on how long the molecules stay attached to the binding site. In fact, the duration of the fluctuation will be, for example, related with the K-off of the molecule from this target site. So the idea is that if you study the fluctuation, you can obtain information regarding how fast the molecule move and how are they moving inside the cell. So Enrico explained you that this information can be obtained through autocorrelation analysis. Just brief overview, because I know that you are learning a lot of new concepts. And so going back to the basic idea has, well, perhaps it helped you to understand these techniques. So the idea is the following. You have in here, this is the autocorrelation function. This is how a fluctuation is defined. And what you do in the autocorrelation function is just multiply the fluctuation at a given point time, times the fluctuation at a given time plus tau, a lag time tau. So what you do is essentially the following. Here, for example, is a region of an intensity trajectory. Here are the fluctuations. And when tau is small, what you do is essentially multiply it by each fluctuation by itself. And you do all the multiplication, you do the average, and this is more or less the autocorrelation function at this tau, at this particular tau. So if tau is small, you can see that you are multiplying essentially the intensity trace by itself, so it will give you a high G value. When tau increase, let's increase it, it's like multiplying the intensity trace by itself, but shifted in a given period of time tau. So now you can see that you are multiplying things that are not completely the same. Now you get, in some cases, positive values or negative values. So in average, you will get a value that is lower or smaller than the one you got before. If tau is higher, what we are multiplying are things that doesn't correlate with each other. So when you do the calculation along the whole intensity trace, what you are gonna get is in average 0. So this is more or less the shape of the correlation curve. If the fluctuations last smaller because molecules move faster through the observation volume, the autocorrelation function will decay earlier. So you can see here that from the analysis of the autocorrelation curve, you can obtain information regarding the mobility of the molecules. So in very simple systems, for example, this one that we studied a long time ago, here it doesn't matter what you have a, let's say, a protein which is fused to a fluorescent protein attached to a lipid bilayer. What you do, what you can do is just propose a model for the motion of the molecules. Here, for example, you can say that the molecules are going to move through two-dimensional diffusion along the membrane. And from the model, you can derive, from the theoretical analysis of the model, you can derive an equation that accounts for the motion. For example, here. So you can fit the equation to the experimental data and obtain relevant parameters of the motion of the molecules. For example, here, the diffusion coefficient. So let's go back. Oh, let me see that. Okay, sorry. The floating middle. Okay, so let's go back to the transcription factor. Okay, what happened when you do this kind of experiments with the transcription factor? Can we obtain information regarding how this transcription factor is moving in the cell? And these are, for example, representative experiments we obtain in these FCS experiments of the GR. Here is just a cell that was transfected with GR TFP. This is a control cell, not stimulated. So this point represents the point where you focus the laser. And this is more or less a correlation curve you obtain in control conditions. Whenever you stimulate the cells with DEXA, you can see that the curve shifts toward a higher tau values, suggesting that the receptor is doing something different when it is stimulated. So the question is, can we obtain information, quantitative information on what the receptor is doing in the air? Of course, we try to do the most easiest thing on the world is trying to fit the experimental data with a random diffusion model. And of course, the data did not follow this model because as the GR is a transcription factor, at least you would expect that it binds and unbinds from target sites in the chromatin. And I had to do a parenthesis in here A long time ago, we were also studying another different transcription factor, in fact, a pluripotency transcription factor in mouse embryos, early embryos. And we got similar curves as the one I showed you for the GR. So here are representative curves we obtained for two populations of the cells in the blastocyst stage. And you can see that this correlation curve presents these long curves. I'm not going to show the experiments now, but we did a lot of experiments, control experiments. We changed the observation volume in the confocal microscope. We analyzed mutants that did not bind DNA. We modified the chromatin condensation by using different drugs. And we did a lot of simulation. And by doing all these control experiments, we reached to a very easy and simple model that could explain the experimental data. In this model, we consider that transcription factor binds to two populations of binding sites. They present what we call the long-lived and short-lived interactions with chromatin targets. And if you analyze this model, you reach to this very easy equation. The correlation equation has three different terms. One that accounts for the diffusion of the transcription factor in the nucleoplasm and the binding to these two populations of target sites. When you feed this the experimental data with this function, you can see that this equation is enough to explain what we get in the autocorrelation experiments. The nice thing about this equation is that you can recover nice information regarding what the transcription factor is doing in the cell. You can recover two important parameters. The first one is the residence time, which is the time the molecule, the transcription factor, stay bound to a given target site. And the second term is the fraction, the bound fraction, which is the relative number of transcription factors that are attached to either the short or the long-lived sites. So we analyze the GR data with this model, and you can see that whenever you stimulate the cell with text, both the residence time and the bound fraction of the receptor to the long-lived site increase when you stimulated the cells, showing that the receptor is attaching for a longer period of time to a higher number of targets on the DNA. So what happened with the monomeric version? Well, you can see here that the monomeric mutant, the autocorrelation curve of the monomeric mutant is more or less the same as the control, as the GR in control cells. And when you do the fitting of this data, what you observe is that both the residence time and the bound fraction are very similar or very close to those measured in control conditions. So these results show that the GR-MON mutant behaves more or less as a wild-type receptor in non-stimulated cells. So let's go to the second question. Can we dissect its interaction, the interaction between the receptor and other biomolecules relevant to its function? And I will show you just one of the experiments we did to answer this question. To do that, I have to introduce a second technique very related to FCS. This is fluorescence cross-correlation spectroscopy. The idea is very similar to that of FCS, but in this case, what you are going to get is the following. You have in here, for example, the glucocorticoid receptor bound to a green fluorescent probe. You have in here a co-regulator that is attached to a red fluorescent probe. And what we are going to do is set the microscope to collect simultaneously the green and the red photons in two different detectors. Okay, so we are going to get the green and red fluctuations in two different detectors. So what is the idea of that? Okay, if this is, for example, in a given moment, the confocal volume, and you get all these species in here, in the green channel, you will get information on the green species. So you get information in the heterodimer and of the green monomers. In the red channel, you will get the information regarding the red monomers and also of the heterodimer, because they also meet in red. Okay, so these are examples of intensity traces you get in the green and in the red channel whenever and heterodimers move in and out of the confocal volume, what you are going to get is simultaneous fluctuation in the green and in the red channel. Whenever you get a green monomer, you only get a fluctuation in the green channel. And the same happens with the red monomer. You get fluctuations in the red and not in the green. So you can see that these simultaneous fluctuations in the green and the red channel contains the information of the heterodimers. How can we obtain information, or how can we extract information from these intensity trajectories? Well, very, very similar to what we did with the autocorrelation function. But in this case, what we are going to do is multiply the fluctuation in channel 1 times the fluctuation shifting in a given period of time tau in the channel 2, yeah, in the red channel. So you can see that the cross-correlation function will be positive whenever you have simultaneous fluctuations in both channels. If you don't have this kind of experiment, of species, sorry, the cross-correlation function will be equal to 0. So we did that with the GR and a co-regulator, a co-activator of the receptor. Here are some results we got in this system. Here is the autocorrelation function in the green and in the red channel, and the cross-correlation. From the analysis of the cross-correlation function, what we can also obtain is information regarding the amount of the relative amount of the heterodimers, and also, if you can see here, from the mobility. But from this analysis of the relative amount of heterodimers, what we can estimate is an apparent association constant. What you can see is that whenever you stimulate the cells with X, the interactions between the receptor and the coactivator increases. If you have the monomeric receptor, the interactions are not as important as in the wild type receptor. You verify that there are indeed impaired interaction between the monomeric receptor with the coactivator. So we could also study these interactions between these two different species. And I'm gonna finish my talk just giving some headlines. Diego talked a little about what happened with the activated receptor. You can see here that the organization of the inactive receptor in the nucleus is more or less homogeneous. Whenever you stimulate the cell, you have a given probability, depending on the specific ligand, of getting these bright dots all over the nucleus. So the question is why the active receptor distributes as this heterogeneous distribution? Is this general for all nuclear receptors? And the most important question any has to do is that important for the biology? And we believe it is important. Let's imagine you have a very simple reaction, A plus B, that gives C. If you have the molecules distributed in an homogeneous environment, you have a certain probability of interaction between A and B. So you have a given speed of the reaction. If these molecules concentrate in a confined region of the nucleus, you get a region that has a high concentration of A and B, so the speed of the reaction in here will go up. Or if, for example, you have in here an inhibitor and the reaction occurs outside the foci, you have a lower concentration of A and B, and the speed of the reaction will go down. So in conclusion, what you can, the relevant thing here is that you can modulate the speed of the reaction by compartmentalization. And compartments in the nucleus are very, very common. You can see here, for example, all the members of the steroid receptor family form this kind of foci. And I don't have time, so I'm gonna show just some headlines of that by doing certain experiments of FCS, scanning FCS, and these cross-correlation experiments, we could determine that these foci formed by an initial interaction of the receptor with the DNA. After this initial interaction, many other co-regulators and molecules relevant to transcription are recruited to this initial seed, and this keeps growing on and on, forming a kind of condensate that has properties compatible with those expected for the for liquid condensates. So just to conclude, many years ago, we thought that to understand the function of transcription factor, we had to have information regarding how the transcription factor interact with its specific site on the DNA. We now know that we also need to know other things about this transcription factor, how it is spatially organized, because the spatial organization will also modulate its function, and how dynamically interact with all different partners in the nuclear landscape. Okay, so I think I'm more or less done. I would like to